Okay, it is 3:01 p.m. here in Abu Dhabi, and let us get started. So, welcome everyone. Welcome to the Space Science webinar series hosted by the Center for Space Science at NYU Abu Dhabi in collaboration with the UAE Space Agency. Today's speaker is Dr. Maria-Paz Zorzano. She is from the Center for Astrobiology, which is a part of the National Institute of Aerospace Technology in Madrid, Spain. Her research focuses on planetary exploration. She's involved with instrument development, astrobiology, and the interaction of the atmosphere and hydrosphere with life. She has worked with several missions with NASA, ESA, Roscosmos, in missions such as MSL, Curiosity rover, and the ExoMars rover. She is the co-PI of the ExoMars Habit instrument, which will evaluate the present-day habitability of Oxyplanum, which is the landing site of the ExoMars mission, which was originally scheduled to launch this year, but it has been postponed to 22. So with that, welcome Dr. Zorzano to our center, and the stage is all yours.
Thank you very much, Dimitra. Thank you very much, everybody, for attending today. I'm very glad to be with you in the Center for Space Science. I'm going to present a talk where we expect at the end to learn how to find life elsewhere beyond Earth. And I call that alien life because it's not from the Earth. So the title is Searching for Life in the Solar System. And before we proceed, let me take my camera off so that you get a better view. I would like to tell you that deep space is not a very nice environment for life. So we would generally describe deep space as a hostile environment. First of all, because the temperatures are extremely low. If you look at the graph on the left, you can see an example of the temperature of the surface of the moon, our satellite. And you can see in blue how the temperatures are oscillating between temperatures that are close to 500 Kelvin and temperatures underneath 200 Kelvins within one lunar day. Now, this thermal contrast is not good for life, but most importantly, life cannot cope with either the lowest limit or the highest limit. Actually, the temperatures about 140 degrees Celsius are sterilizing for life. Not only temperatures are really non-friendly for life, as we know on Earth, but also when you're in deep space, you're exposed to space radiation. In the case of the solar system, you have solar radiation, but also you have galactic cosmic rays. So you have other sources from beyond the solar system. And it's natural to think that this will happen in any other environment around a star or floating in the environment between solar systems. Here you have on the right a graph of the measured radiation dose observed by the Curiosity rover on its traverse to Mars. And you can see the red graph represents what you would receive if you're 500 days flying around in space. And you can compare it with what we receive on the surface of the Earth, our planet, our super habitable planet. You can see it in blue. And as you see, there are orders of magnitude of difference. And radiation is indeed one of the most damaging conditions for life. It's strongly sterilizing. So these two pieces of environmental information alone just tell you that not everything is a place where life can survive, but maybe within rocks, maybe within asteroids, or maybe deep within the moon, you can have remnants of life, or you can have some molecules that were important for the origin of life, and they are preserved from radiation. The rocks are screening radiation. But those environments are not the places that we consider habitable, meaning life cannot thrive there. You cannot have one cell and have it holding metabolic activity or reproducing. There have been some very famous programs for the search of alien life, as we think of intelligent life forms elsewhere, deep in the universe. One of the most important examples is this radio telescope array that is dedicated to traditional astronomical observations, but was also used as part of the SETI program, the Search for Extraterrestrial Intelligence. I think it's nice to look at this because it represents a very long time effort of search of artificial signatures that may try to demonstrate the existence of a possible civilization communicating in an artificial way. This, so far, to my knowledge, is the best example or the best candidate of what it's a signal that has not been understood scientifically, but it still is only a very, very faint indicator. It's a narrow band signal that was detected at the Ohio State University Observatory. And as you see, the observer wrote wow, where this flash of very high intensity was detected. And that is why the signal is called the wow detection. But actually, this is not a true demonstration of life. If you want to find demonstrations for life elsewhere, you really need to have unquestionable observations that can be reproduced by independent observers and understood only with this explanation. There are, however, other attempts to look for life elsewhere. And the more traditional approach is first to find planets, to find habitable environments. The reason why we're searching for planets is because, as we said before, anything that is exposed to deep space is going to be exposed to radiation mostly, and it's going to have no atmosphere, so it cannot hold liquid water. So our first assumption is that in order to have life, you need to have a planet with an atmosphere. And in the search for exoplanets, that is any planet orbiting other star that is not ours, we have different platforms that are dedicated to this endeavor, and one of them is the Kepler Observatory. And when you add all the efforts of all the scientists together, you can use different methods. One of the most important ones is the observation of transits. You can see in this artistic impression an example of a planet as a black dot orbiting a star. And if the orbiting plane of the planet is within our field of view, every now and then, in a periodic way, you're going to see reduction in the brightness of the star, and that is called a transit. And by monitoring that, we have a very, very big number of planets that have been discovered. As you know, this is a graph of the evolution over time of the number of planets that have been detected. Those that have been detected by transits are marked in green, but there are other technologies that are very well documented and used extensively by different institutions. The information that this graph tells us is that obviously there are many, many, many exoplanets in the universe. However, when we want to narrow down the search for habitable environments, we need to apply one condition. And the condition we apply is that we know that life on Earth needs liquid water. So when we screen this list of planets and we put the condition that water, if detected, may be liquid in that environment, then this search is reduced to only about 30 planets. So the principle that we have to keep in mind in our search is that first we will find habitable environments, and then we will try to find some signatures of life. Now we're going to focus on our solar system because as I said, looking for life elsewhere in the universe can only be done by looking at planets and looking at the atmospheres and hoping that we can see some signatures in the atmosphere. But in our solar system, we can have other approaches because we can build missions and get closer to the objects of exploration. So just as a first remark, remember that today we're not looking for intelligent life in our solar system. We're just looking for microbial life, okay? Now life could be still living, this is what we call extant, but it could be extinct. We are also looking for remnants or footprints of life that no longer exist on an environment. We can only take as reference what we know from our planet because to date is the only single point of habitability that we know in the universe. And what researchers do, especially when you tackle this with the perspective of astrobiology, is that we investigate all the environments where life has appeared ever, all the extreme conditions that life can tolerate. And by looking at these big margins, let's say pH, temperature, pressure, nutrition, radiation exposure. Then we have our habitability condition. And then with that, we can check the planets and satellites that we are exploring. One important remark, however, is that life as we know on Earth has co-evolved with our planet. Our planet has an evolving history. It has changed, for instance, dramatically the atmosphere, the composition of the atmosphere. Originally, we did not have oxygen, and now we do have oxygen because of life. And not only we have oxygen, but also we have ozone. And ozone is produced by the interaction of ultraviolet radiation with oxygen. And the good thing about ozone is that it protects, it screens UV radiation. So it protects the surface from very strongly sterilizing radiation from our star. And that has allowed life to go above to the surface. So as you see, all the memory of our genetic information is mixed with the memory of the planet that we live in. And also the planet has changed, the minerals have changed, they have been oxidized by the oxygen that was released by life. So this coevolution may have somehow biased also our view of what is life. And we should be open to the fact that if life has appeared in another planet, let's say that life traveled from Earth to Mars at one point, then as Mars had a different evolution, life should have adapted to that different evolution and maybe slightly different from the one we know. So that said, the field of research that tries to tackle this in a comprehensive way is astrobiology. Most of the search for life that we do is based on exploring our own planet. So we learn a lot by doing field campaigns to extreme environments. We learn a lot by doing simulations in our laboratories, by creating chambers that can simulate the environment of another place. And then we compare that with the observations of spacecrafts like orbiters or landers or telescopes that are studying other objects of the solar system. And of course, we compare that with models. Models are very important here. Let's say we find an environment that we think it is or it was habitable. What should we search for? If you really want to tell or convince the community that there was life there, you need to find what is called biosignatures. And biosignatures can have different forms. You can have biosignatures that are operating at the atomic or element level. And the most important examples of this kind are the alterations in the redox state of any composition of a mineral, of a fluid. All the life that we know is based on the transfer of electrons. All the metabolic activity that we do is using transfer of electrons. So this alters the redox conditions of the environment. And it also requires that there is some kind of gradient of redox in an environment. When all life forms perform their routinely metabolic activities, the elements that made the molecules that we use, most importantly, carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphor, change in their isotopic composition. Generally, life tends to favor the lighter isotope, especially with carbon. And when we study this on Earth, we can very clearly distinguish where carbon comes from a abiotic source or where carbon has been recycled through life. So if we found those kind of signatures in a sample, it could be a good indicator of life. There are other indicators that can be seen from a distance, like gas. Certain volatiles are released by metabolic activity. We were talking before about the emission of oxygen as the one we can think of. On Earth, for instance, just through photosynthesis, every plant, every algae is releasing oxygen to the atmosphere and fixing CO2. And if you look at our own planet, here you have a graph of the monitorization of oxygen and CO2 over the years. And if a scientist from another world look at our planet and had a look at this graph, they could say that they think there is something breathing at a large scale in this planet. So this gas may be considered as a biomarker. We have also seen biomarker that is very interesting on Mars. We have to detect methane. Methane on Earth is produced most generally by life, but there are other sources that are not related to life. Also, oxygen and CO2 can be released and captured by other sources that are abiotic. So this gas by itself is not a biomarker. It could be really nice if I have access to a sample from another environment, and if I found a molecule that contains genetic information or any kind of information, like a protein or DNA or RNA or fragments of these molecules, because these molecules are very easily broken up, then I could say that that looks like a life signature. Also, you find lipids, because lipids are super essential for life. We need them to form vesicles and to separate the inside world from the outside world. Or if you found molecules that had very specific functions, like chlorophyll, that could be, again, a very strong indicator of life. Some of these molecules, like amino acids, can be produced in the lab in abiotic conditions. There is a famous Miller experiment that can produce some amino acids. But to date, we don't have a process that can produce all this variety of very intelligent molecules that are specifically related with functions that are critical for life. Now, also at mineral level, so a larger scale, you can find biomorphs. You can find structures that look like a fossil, a remnant of something that was related to the structure of your organism. Now, biomorphs are very tempting because to our eyes, they look like structures that have been produced by life. But in reality, we know that you can have those structures formed also abiotically. So the approach that scientists like combines the detection of multiple of these biosignatures simultaneously in one sample or one object of study. Now, you can imagine that if we want to have access to this information beyond the solar system, it's completely out of the question. And it's already pretty difficult to have access to this information on any object of the solar system. But this is the strategy, this is the roadmap that we are following. Let's take one example. In my mind, the most habitable environment that exists within our reach is Mars, besides Earth. Here, I want you to look at our two rocky planets. Both planets have an atmosphere. Both planets have water or had water in an early time, as it happened on Mars. And if we look at the history of coevolution of Earth with life, and this is a very nice graph of the Earth's biogeological clock, you can start on the origin of Earth, and as you move rightwise, you can see that life appears about 4 billion years ago, and we start having phototrophic bacteria and cyanobacteria producing oxygen close to 3 billion years ago, and then life evolves into more complex life forms. And at that time, at this 3.5 billion years ago, Mars looked like that. It had an ocean, it had rivers, it had water with different compositions. We had minerals with different redox gradients. And from what we know now, there were many ingredients that were important for life to survive, at least. So we would say that Mars was habitable, and therefore we had two planets. One was inhabited and the other one was habitable. So it's possible to think that there may have been some transfer of matter through asteroids from one planet to another. As we do have fact, we have meteorites from Mars on Earth, so it may have happened the other way around. If we want to look for life on Mars or elsewhere, the first thing we need to know is that we need to find environments that preserve those precious signatures that we talked about before. And they're environments that are good preservers that I have marked here in green, and they're conditions that are very bad to preserve life that I have marked here in red. Generally, if you have, let's say we want to conserve a protein or a lipid, maybe a long chain lipid, then the best condition for its preservation would be to have very rapid sequestration into a matrix that is going to protect that sample. And that would be a rock that contains silica or carbonates or shales or evaporitic environments. It's good if the conditions where this molecule was preserved are reducing. life doesn't like oxidating conditions, then those molecules could break apart. And it's also good if through the evolution of those samples, the samples have been evolved to low temperatures and low pressures after burial. On the other side, if those samples have been exposed to oxidation, radiation, as we said before, and have been exposed to elevated temperatures and pressures, and that has transformed the rock into a metamorphic rock, then the chances to have these preserved biosignatures are lower. There's some elements that may still survive, like isotopic composition, but all the other elements that we're really looking for may be completely destroyed. So this kind of a very simple memory card is what Mars explorers have in their mind when they select the samplings, the landing sites of the missions. We said before that the isotopic composition is a clear indicator of the presence of life. And I want you to look at these graphs. Unless you are an expert in isotopic analysis, it may look to you very strange. But overall, this is telling you, the graph on the left is a look into the past history of Earth. If we go to the 3.5 billion years that we talked about before, can you look at the red dots? Can you see that it goes towards negative delta 13 carbon? That indicates that life already at the time tried to have access to the lighter isotope of carbon. And when those life forms were trapped, that local environment of the rock was enriched in that isotope, okay? There are many studies looking for the oldest samples on Earth that try to add points to this history. So if we ever get samples from extraterrestrial origin that have a high concentration of organics, and we look and we see some isotopic trend, then that would be a good indicator for the presence of life. However, we know that organics are everywhere in the universe. They're even floating in the universe. We can detect them with telescopes. And we know from multiple missions that have investigated, for instance, meteorites, or have investigated asteroids or comets, that organics are preserved and formed within ices. So when we look at organics from meteorites, if you give it to a chemical expert, this expert is going to find a lot of products. And there's only one way to distinguish if those products were belonging to a life signature or were produced abiotically. If the organics were produced in a non-life process, then most generally you expect to have complete isomeric diversity. You expect to have racemic composition. That means you have both levo and dextro, for instance, amino acids or other products. Both chiralities would be present. You would expect to have a soft decrease in abundance of molecules with increasing carbon number. So when you produce things in the lab, things are produced sort of statistically, and the longer chains are less produced and the shorter ones are more produced. Very rare in these experiments, you would find unique forms. You would find a broad distribution of molecules with no specific functionality. You would have positive enrichment of carbon-13, and you can interpret how these products are produced one from another through stepwise synthesis. Now, if you gave this very clever chemist a sample that comes from life, what this person would find, on the contrary, could be specific isomers, predominance of one chiral form, ideally homochirality, abundance of molecules that have one specific function in biology. You would find some patterns of common forms that cannot be produced stepwise, but require biosynthesis. And we would find carbon-13 depletion. So this is like a general recipe for the chemists working in the lab about diversity in the organic molecules that you may expect. Going backwards one step, we said before that it's very important to find habitable environments. So we cannot apply this very detailed analysis to anything we get from space. We have to first focus our energy on objects that are habitable. And I've taken this picture from Interstellar. I don't know if you have seen the movie, but in this movie, one of the starting points of the story is that humankind is looking for habitable environments elsewhere. And the question of if a place is habitable, it's really hard to answer, but we take as reference our own planet. So if I had to paint from scratch a perfect planet, I would, as I said before, ask for liquid water. And therefore, I would ask for conditions that can allow for liquid water to exist. Ideally, an atmosphere or a crust of ice that creates some pressure. I would also need some energy source, probably from the sun, but also from other redox gradients within the constituents of the planet. I would ask to have access to carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphor. And that is not so trivial. Especially nitrogen, generally it's stable in a molecule, in a volatile molecule, N2, but it's very hard to break apart that bond and fix it to the ground. And I would like to have a protecting environment that protects me from UV radiation and space radiation, and also that allows for some kind of recycling or climate or weather that moves things around and allows to have some mixing. So this is our planet, and it looks super nice. It's a super habitable environment, but where else do we have something that may remotely look like this? Well, we're lucky that in the solar system, we have 4 good candidates that are not exactly like Earth, but they have some similitudes. As I said, my favorite candidate is Mars. Mars, we know it has water. It's frozen in the polar caps, and it's also as a volatile in the atmosphere, but we know the atmosphere escaped, and most of the liquid water that was on the surface has gone elsewhere. We also are investigating the habitability of Europa, a frozen satellite of Jupiter, and we believe that underneath the ice layers of Europa, there may be an ocean, and those oceans are considered as habitable environments. And the same applies to Enceladus. We talk nowadays about icy walls as potential environments where life could thrive. And another candidate that is very important for astrobiology is Titan. Titan doesn't look like a super habitable environment, but it's full of hydrocarbons, and it may have taken a very important role in the origin of life, because hydrocarbons and all organic molecules are very important for life. And lately, there have been some indications that there may be water in Titan too. So these are the main exploration targets for habitability, to my knowledge. When you explore an environment, if you take this task very seriously, you need to launch a full family of orbiters and eventually landers. And much better if you have rovers, because that gives you some mobility and you can bring your analytical tools to different places on the planet and compare. And this is the case of Mars. And as you know, the Mars exploration program started a long time ago. In this graph, you see the success to non-success ratio. The red missions are those that did not succeed, and the blue ones are the ones that are successful. And you, in Emirates, you are also running now an orbiter to continue to investigate the atmosphere of Mars. And these pieces of information are adding together. And we are now in a better position than ever to understand how Mars was and how Mars may be right now. But in order to answer the question about life, the most important observations are those that have been provided from the surface. And this here is a map of all the landers and rovers that have ever reached the surface of Mars. Not all of them reached the surface in a successful way. So I'm just going to focus on those that are now operating. And as you may know, the Curiosity rover has been operating from the surface of Mars from the year 2012. We also have the InSight mission. This is not strictly related to habitability, but it's also giving information or trying to give information about the inside of the planet. This is a lander. We have the ExoMars mission, a European mission that will be launched in 2022. And we have the Mars 2020 rover, which is on its way now to Mars. In addition to these missions, we had already in the 70s, the Viking landers. The Viking 1 and Viking 2 were very ambitious missions. And they were trying to give answers to the question of life. And we also had the spirit and opportunity to small rovers with solar panels that were exploring the surface of Mars for a long time. As I said before, the Viking missions were the first ones that seriously tried to answer the question of habitability from the surface of Mars. Here you have an image of Carl Sagan with a replica, a real-size replica of the Viking lander. And unfortunately, even though this mission was super successful in many objectives, we did not get enough information from the analytical instrumentation to assess without any shadow for doubt if this environment hold life. The measurements of the Viking lander campaign have been revisited afterwards with what we know from other missions, and still today there is no consensus in the scientific community. Nevertheless, these were pioneering observations, and it was the standards of this mission was so good, so high, that in many aspects it has been mimicked afterwards with the following surface exploration missions. The one mission that has been dedicated with all its heart to the quest of habitability is the Curiosity rover. You can see here the rover and the mount on its back. This is a selfie. We know from this rover a lot about Gale crater, where it landed. This is a location near the equator. And as I said, the scientific objective of this mission was to determine if Mars was habitable. And spoiler alert, this is what we found after a few years. We're continuing to create this image of how Mars looked in the past. This is an artistic representation of Gale crater. And adding the observation of multiple instrumentation on this rover, we concluded that Gale crater was habitable, precisely at the time that I told you before that life was inhabited. And we concluded that because we observed that there were conditions for liquid water, that there were redox gradients, that there were essential elements like nitrogen that was available within the regolith of Mars. So from this point, we can extrapolate to other sites of Mars, and we think we can clearly say now that Mars was habitable in the past, and it had a very thick atmosphere, or at least thicker than the one we have now. We also have learned through the observations of Curiosity as it moved along its track. When we drill a few samples of the surface, the upper few centimeters at different spots, we have found organics preserved in this environment. Now, the organics that we have found are not those super exciting biomolecules that we were talking about before, but they're still very, very important because they indicate that they can be preserved within these rocks. And we were worried that the organics may be broken up by radiation and stresses. So the fact that we found them is very interesting. We cannot tell so far if they were produced in situ or if they came from other environments. The truth is that the instrumentation that the Curiosity rover has is not dedicated to finding other kind of molecules like amino acids or sugars or lipids. So it's just precisely designed to find these kind of molecules. So the fact that we didn't find those more interesting molecules doesn't preclude its existence. We wouldn't expect them, however, at this very shallow depths, because as I said before, these environments are strongly irradiated. We have also found that these organic carbons are combined with chlorine, and this may be part of the post-processing that the samples had to go afterwards, or it may have happened naturally during its formation. But the truth is that the samples have been preserved in 3 billion years old samples. So this is a good record. Altogether, with this kind of rovers, we know that very big environments or large environments of Mars have contained sedimentary environments where we may sample organics or leftovers from life. And we know that the rocks of the surface of Mars have 3.5 billion years old, and we're worried about this because on Earth, we didn't find so far any biomolecule that is longer than 1.6 billion years old. And that is in our planet where we have this shielding atmosphere. So if the moment where we think Mars was habitable is at 3.5 billion years ago, what are we going to find after such a long time? There have been some proposals of what are the environments of Mars where biomolecules may be better preserved, or what are the environments that guarantee that something interesting is going to happen. And I particularly like this proposal by Steve Ruff and Kathy Campbell and colleagues. Based on the observations from the left, on the left is a picture from the Spirit rover at Columbia Hills on Mars. And these structures are what is left after the existence of a hydrothermal environment on the surface. You have an example on the right of El Tatio, the hydrothermal field in Chile. And these structures are very similar. And why were these researchers interested in these specific environments? Because we know that hydrothermal vents are very important for life, for the emergence of life, but also they are very good at sequestering life forms and biomarkers. So they can be used to detect leftovers from life. And I'm going to give you an example precisely of El Tatio. And this is to illustrate why field campaigns on Earth are critical to our knowledge. This work has been done by some colleagues of mine. They went to Chile and they visited this place, and you have in images D, E, and F examples of big structures of hydrothermal mounds in different stages of the evolution. On the left, you have a liquid mound. In the center, you have a steam mound. And at the right, you have a dry mound. And my colleagues took samples of these three hydrothermal places, and they could find all kinds of living organisms contained with them. However, they were slightly different because, as we said before, life forms evolve with the environment where they are, or they are selected together with their environment. And they applied a number of techniques that can only be done here on Earth in the lab, applying standard micromolecular techniques that are very time-consuming and very specific. They could find, for instance, that if you look at this color coding on the right side, on the dry column, you would find what is very present in the dry mound. And I want you to pay attention to that one. The one on the right tells you that you can find proteins after some time in this dry structure that indicate that whoever was living there survived hydric stress and also had some synthesis for carbon storage. You can also find that these living organisms were most importantly or most predominantly bacteria that were reducing perchlorates. And this is important because we have found perchlorates on Mars. And also they have some specific methods to adapt to sulfur oxidation. This is a kind of a study we would really like to perform on a sample from extraterrestrial origin. This is more from that work. And if you compare again the left, center, and right, the three types of mounds, I want you to pay attention to the geochemistry and biomass studies. And again, we find this carbon isotope negative footprint that is indicative of life. You could perform all kinds of very specific studies, including lipids. I told you that they are very important, and these molecules have very specific distribution when they have been produced by life. And you can infer a lot about the communities that were living there. But the problem with this is that if we go now to Mars, and if you go now to one hydrothermal vent, it will never be in liquid or steam mound or even in the dry state. It will have passed 3.5 billion years after this latest picture. So whatever is there contained or preserved, if there was life, is going to be strongly damaged. And we have to be ready to understand those kind of measurements. There is one mission that is going to go one step beyond, and this is the ExoMars Curiosity rover and surface platform that will be launched in the year 2022. And this is not trying to find if Mars was habitable, which was the goal of the Curiosity rover, but it's going to try to find biosignatures. And because we know they need preserving environment, the rover has a drill that is going to try to analyze samples from the 2 meters underneath in the surface, because at that depth, we know radiation is strongly screened. So these observations will be very, very important to understanding of life, if it existed, if it adapted, if it was preserved. But if it has, of course, again, limited capabilities, because the instrumentation that you can put on board of one of these spacecrafts is very limited. The landing site of ExoMars is Oxyaplanum, and this is a region that is very flat, that facilitates the landing, and it's a region that is very rich in clays, and we know clays are important to preserve live biosignatures. As we said before, there is another mission on the way. This is a Mars 2020 rover mission, and this mission is similar in many ways to the Curiosity rover. It has some additional payloads. Some of them will be very exciting, like the new helicopter, the drone. But it has one specific capability that is very important to the studies of life. And this capability is the capability to make samples, store them, and leave them on the surface of Mars. And this is the first step of a larger scale architecture, an agreement between NASA and ESA. And this is part of the Mars Sample Return campaign that will take within a decade at least. It will take a long time, but we're just observing now the first steps. Once these samples have been taken, they will be left on the surface, and eventually in a few years from now, a lander by the European Space Agency will go there, fetch those samples, and bring them back to Earth. Here you have an overview of Jet Zero Crater, the place where we are landing the Mars 2020 rover. It looks very habitable to our bare eyes because it looks like the delta of a river. So we expect it to have minerals that, well, we do have minerals, we know that from remote sensing, that indicate the presence of water, but also we have structures that indicate that there is deposition and sedimentation, and this is a good place to look for. Clays and carbonates are good environments for preservation of biosignatures. And on the right, you have a schematic representation of this full architecture of the Mars Sample Return Program. We are now on the first stage. The Mars 2020 rover is going to Jet Zero. It will land, catch the samples, leave them there, waiting for a while, and then another mission sent from the European Space Agency will go, land, have a small rover fetch the samples, bring them back to an orbiter, make the rendezvous, and bring them back to Earth. And once we have them on Earth, we want to do all kind of analysis. Many of these analyses are related to other questions about the history of Mars, the evolution of the atmosphere, but some of them are also related to life. And when we do that study, we will use the techniques and technologies that are applied on Earth when we investigate the origins of life. There are other missions that will bring also interesting information to add to this question. Those, in general, all those sample return missions will be very informative to us. There is right now a super interesting mission going on, the OSIRIS-REx by NASA, that has caught a significant sample from the surface of the Bennu asteroid. We also have on its way now the Hayabusa 2 mission, also sampling from another asteroid, Ryugu, and we had before the Hayabusa 1. This is by the Japanese Space Agency. And both sample return missions will provide us critical information on everything, including how organics are preserved in these environments, what techniques we have to use, how we can preserve cross-contamination. There is another mission that will sample from an environment that is near Mars, and this is the MMX mission by JAXA also. It will return samples from Phobos, Deimos, the satellites, the natural satellites orbiting Mars. That said, those will be missions that will give us information about sample return, but we have other missions that are very focused on investigating the habitability of those environments that I told you about before. We have the NASA Europa Clipper that will investigate Europa. We have the Cassini mission in the past that found organics indirectly by remote sensing on Enceladus. We observe the plumes, we observe remote sensing observations through infrared spectra and find the signatures of very interesting molecules. And we have in the future the Titan Dragonfly drone that will try to get inside Titan to the surface and investigate this super interesting atmosphere where you have rain of methane, organics, and an atmosphere that is very rich in nitrogen. When we implement these missions of exploration with orbiters or landers or drones, it's very important that as long as we're investigating life, we preserve pristine the environment. And the policies that describe this are called planetary protection policies. They basically tell you that when you are visiting an alien land, before you cross the border, you have to ask yourself, am I bringing what I am allowed to bring in? Should I declare it? Should I just take it away from before I cross the border? And this was described when many countries joined together a few decades ago, and they declared the International Space Law Treaties. And as part of those treaties, the planetary protection policies were implemented, starting or based on those treaties. Basically, this protection policy are applied by space agencies like NASA, ESA, JAXA, and all the new space agencies that are taking part in this endeavor. The agreement that are undertaking when you do this is that you will try to preserve cross-contamination from potentially habitable planets and moons. And the way to do this basically is applying what already started with the Viking mission. If you're sending a lander to an environment, you have to sterilize it and clean it and demonstrate that you're not bringing any bioburden. You're not bringing spores, you're not bringing uncontrolled amounts of organics. And if you are bringing some material to the moon, let's say, at least you're making a list or a declaration of what you're bringing so that other scientists will not be confused later on if they found those signatures within their research. So to sum up, in our search for alien life, we have learned that first we have to locate a habitable environment. We cannot just put our instruments searching for life everywhere in the universe. We can narrow down our targets of investigation by using remote sensing observations, modeling, and so forth. And once you know where to look for, you are going to prepare methods on Earth with analog studies or with laboratory studies of what kind of biosignatures we would be looking for. Very important, as you do normally on Earth, you have to avoid false positive. So you have to avoid cross-contamination. And the same applies when you go to other worlds. And this is what we call planetary protection. Discovering past or present alien life forms could be such an amazing result that we really need unequivocal proof. It would have impacts on humankind in different fields. Impossible to foresee what other fields beyond science, but for sure it would have philosophical impact too. Even if we have a negative result on life detection, even if we conclude that we are alone in the universe, it would have a very important impact in our understanding of our role in the universe. So we have to make sure that this question is answered in a clear scientific way and that all nations can contribute to this investigation. And with that, I close my presentation and I'm open to any question. Thank you very much for your attention.
All right, thanks a lot, Maria. Yeah, that was a great presentation. Now let us see if we have any questions. Yeah, we have a question from an anonymous attendee. What are your views on the right time for human exploration of Mars?
Well, I think we're very far away from taking that mission seriously. First of all, because in order to bring a human to Mars, we need to be sure that we can bring that human back to Earth. So the first step to demonstrate that is to bring samples from Mars. If you bring a rock back, then I can believe we can bring a human. The second problem is that as soon as we bring humans to Mars, unavoidably, we will also bring our own bioburden. We live together with an enormous amount of organisms. They actually are about 2 orders of magnitude more than our own cells. So as soon as any human activity starts on the moon, it's impossible to avoid cross-contamination. So by then, we should be sure, we should have closed the question about natural life forms from Mars. And those missions are just starting now. The third reason why I think we are not ready yet, it's because we don't have the technology to produce the propellants and the human needed resources like oxygen and water in situ. We need to demonstrate in situ resource utilization capabilities because the amount of propellant that we need to lift off from Mars is so big that we cannot send a spacecraft from Earth that has the propellant to get away from the gravity of Earth and later on get away from the gravity of Mars. Now that said, we have the tools to make a roadmap, and many industries and many space agencies are working consistently to get to this goal. So I think once we have the Mars Sample Return Program closed and we have analyzed those samples, we will be somewhere at midway to the preparation of the human exploration of Mars.
Hi, Maria, this is Srini. I'm sorry I was late. Thank you very much for a very interesting talk and a very good answer you just gave. Thank you.
You're very welcome.
Yeah, so I have a question. So when we talk about detecting biosignatures, especially in case of exoplanets, a lot of work has been done. But recently, as you know, the detection of phosphine, it's a planet next door and it is so controversial. So what do you think about then, observational signatures of life in the solar system or beyond?
Yeah, that's also a very good question and very timely. Well, this is the way to work. You have to look at remote sensing observation. You have to find absorption signatures that indicate the presence of a molecule. And this is what this article reports. Other researchers do not find the same. But anyway, anywhere in the solar system, when you find an interesting absorption signature, let's say everyone agrees that that molecule is there. Phosphine is not a life-critical molecule. It's important because it demonstrates, if it's there, it would demonstrate the presence of phosphor, and that is an ingredient that is required for life. But it's not a product that can only be produced by life, and it wouldn't be one of those essential molecules that we're looking for. However, any activity that we do on exoplanets will always be this kind of detection. If we find phosphine on an exoplanet and water and oxygen, we will still have absorption bands that are very faint, and there may be processes that are not related to life that can produce them. This doesn't prevent us from doing this research. This is precisely the kind of research we have to do with the techniques we have at hand. But we have to be very cautious when we jump from detection of volatiles to making a direct connection with life. And in the case of Venus, I think this connection is really, really risky.
Yeah, true. And every day we see some new research, and yeah, it is becoming more interesting every day.
Yeah.
We have another question. Yes, another question is, what is your take on space mining, especially on asteroids?
Yeah. Well, it's one of those steps that we talked about before. In situ resource utilization needs to be demonstrated in space. Somebody has to try and demonstrate it, even at a smaller scale. But I think there are some regions that still need to be clarified, especially with regard to the outer space treaties and who owns what when you go away from Earth. I don't think there is a clear agreement internationally about that. But independently from that, there will be at some point agreed upon nations with one-to-one agreements or with global treaties as the one of the UN. Then still we have a technical question. And if you have seen how difficult it is to sample from an asteroid, the OSIRIS-REx mission or Hayabusa missions, it's extremely difficult. It's even more difficult if you have to drill. We have now the InSight lander trying to drill on Mars, and it's super hard. When you're drilling a material that you don't know how it behaves, many times you're not just not going to get your drill out, or it's going to be super frozen if it's the case of an asteroid with water, like in the case of the moon. So before we get to mining, we have to demonstrate that the tools that are so easy to use on Earth, like drillers and stuff like that, can work there. Even imagine what it would be like to extract material in an environment with such a reduced gravity. It's easier on Earth, but if you extract something floating around an asteroid, you really need to think in a clever way how to capture that. Now, the fact that it's difficult doesn't mean that it's not possible. It just means that you have to make a plan. And first, all the steps are called demonstrations. So we will see in the next decade how to approach an asteroid, how to land safely. That is super risky, again, because of the gravity. It's very risky to control your position in an object that has such a very reduced gravity and is moving and is rotating. So we have to demonstrate that we can land, we have to demonstrate that we can drill, that we can manipulate products, and then demonstrate that it's energetically interesting to do all these processes there. Because if it's so costful to go there and come back, then that will never be mining. It will still be a demonstration phase. That said, this is what we have been doing for centuries. Humankind is always exploring the environment and developing tools that allow us to explore more. So there will be a moment in the future, maybe the first mining will not be focused on minerals, but will be focused on water. Water is a precious value in space. And there are now ongoing plans to visit the moon and demonstrate the extraction of water from the moon. So to me, that will be the first mining activity that we'll be seeing in space.
All right. Yeah, the drill from the ExoMars mission, a replica is going to be displayed in Dubai next year. I read about it somewhere.
Cool.
All right. If there are no more questions, then let us once again thank the speaker. It was a great talk, and see you everyone next week. Bye-bye.
Bye.
Goodbye.
Thanks a lot. Thank you, Maria.
Thank you very much.